Hello, everyone. Thank you for tuning in. I'm Kevin Johnson, a research scientist at the Jackson Laboratory, and today I'm excited to be here to tell you about our new OPTIMUM project, which stands for Optimizing Engagement in Discovery of Molecular Evolution of Low-Grade Glioma. And that's a mouthful, so we'll stick with OPTIMUM. And OPTIMUM is a subset of a larger study known as the International Low-Grade Glioma Registry. And this registry aims to understand or better understand how treatment affects patients. And the particular disease that we'll be talking about today is glioma. So we'll provide a little bit of background on that, and we'll talk about some of our research, and then also go into this new project that we're really excited about. So what do we know about glioma already? Well, we know it's the most common malignant brain tumor in adults, and that the precursor for these tumor cells are also the precursors for normal glial cells, such as astrocytes and oligodendrocytes. And these cells are cells that normally perform supportive functions in the brain. So they really assist neurons. And this larger category of glioma can actually be broken down a little bit further into based on the IDH mutation, which is a specific mutation in the tumor cells of these cancers. And that is those that have the IDH mutation, so IDH mutant tumor, and those that do not, the IDH wild type. And there's both clinical and molecular differences between these two tumor types. The IDH wild type tumor tends to be a higher grade, so more aggressive cancer cells. These are also diagnosed in an older patient population. We can contrast that with the IDH mutant tumors, which are typically diagnosed at a lower grade, more grades 2 or 3, and also in a younger patient population. And so we're going to focus today mostly on these low-grade gliomas, which tend to be made up mostly of IDH mutant tumors. So how has this been studied in the past? What type of approaches? Well, one such way is through this creation of the low-grade glioma registry, which one of the leaders on the OPTIMUM project founded. And to be a part of this registry, what has to happen is that patients have to have a specific diagnosis to be included, but they upload their data, they share their data with the registry, and so that the registry can begin to analyze the data in such a way that helps us understand how these patients respond to therapy. And so what do you need to be in this registry? Well, you have to have a diagnosis of a low-grade glioma. So that is an LGG. And this we can think of as from a histological point of view of having a tumor that's been referred to as an astrocytoma, an oligoastrocytoma, an oligodendroglioma, and a mixed glioma, depending on the year in which you were diagnosed, as these names have changed a little bit over the years. To be in the study, you also have to have a year of diagnosis in terms of your age, between 21 and 79, with a confirmed pathology report that says grade 2 or grade 3 to better help us understand this relatively rare disease across many patients worldwide. And there are no restrictions with respect to sex, gender, race, your date of diagnosis, or the place of residence. It's an international registry. And again, the goal is to better understand how these patients respond to treatment. Why do we even need a registry? Aren't there other studies that can be done? Well, traditional epidemiological studies have some drawbacks, and this includes a high cost to conduct the study, but also these can be very time consuming for patients. And unfortunately, there are some studies that actually require healthcare permissions. So sometimes patients are excluded without them even knowing about it. So there's really a need for new methods to build cohorts in which we can leverage different people's experiences worldwide to study a relatively rare disease. And one of these methods is to create a registry and leverage our stronger social networks, so social media. And this can be through either texting with friends, it's actually blocked off here, but saying, hey, have you heard about this registry? Do you know anything about it? Would you like to join? Give some more information. And so communication can happen between friends. It can also happen online through national organizations, and also through maybe fellow patients or patient advocates. So this low-grade glioma registry, it really allows us to perform sort of a grassroots effort in helping patients be engaged in research, and also helping us as researchers understand how treatments affect these patients and their disease. So what else can we do to study these tumors? Well, we know that these tumors sometimes recur over time. So where I come into the picture is I'm a genomics researcher. And what that means is we study the tumor samples and look at the DNA to see where the specific defects are in DNA to better understand why these tumors occurred in the first place and how they might be treated based on this molecular profile. So what we typically do is we've studied large patient populations, maybe at their initial tumor, to understand what happened there. But our group is particularly focused on how do these tumors really evade these therapies? How do they, in some patients, these alterations lead to a recurrence? And so what we do is we study the tumor at two different time points of the initial disease and recurrence disease, and then look at how the patients were treated. So maybe, for example, some of the treatments include observation, where we obviously wouldn't have a sample. Others include surgery, chemotherapy, and radiotherapy that may occur in this time in between these two samples, these two specimens in which a patient's surgery has enabled. And so what we've done is similar to this low-grade glioma registry, is we've conducted a grassroots effort where we've worked with hospitals around the world, shown here in this map, to form the Glioma Longitudinal Analysis Consortium, or GLASS Consortium. And what we can do is we can study those patient samples at two different time points to understand how those molecular changes that occur between the two may be causing their tumor to come back even after the initial therapy. And this project, among many others, is led by OPTIMUM leader Roel Verhoek, one of the leaders, as well as myself. And our purpose is really to study what are the genetic changes that occur over time in these tumors and in the face of therapy. However, this has mostly occurred in high-grade gliomas, the IDH wild type that I discussed earlier. So there's really a need to also study this in IDH mutant tumors. You can see some of our recent publications in different journals over the past few years if you would like to learn a little bit more about the scientific details that we've found by studying these 381 patient samples. But maybe I'll give you a little bit of an overview now to just give you a taste of what this research looks like when it happens for real. So one of the things that we look for when we're doing this is, well, what are the changes that we ultimately want to study? And with molecular data, there's many different types, but I'll just cover a few here. And these include mutations, which you've probably heard about. And these are changes to the DNA code that result in different proteins that change the way a cell behaves. And this can make a cell more resistant to therapy, can make a cell more aggressive. So we studied the patterns of those mutations in tumor cells from a patient sample. We also study copy number changes, which is the differences in the gene numbers, and when they're out of balance can also cause tumors to proliferate uncontrollably. Also, the tumor clonality, which is really just to say there's different populations of cancer cells, and some cancer cells may harbor some type of genetic defect, and another area of the tumor may harbor others. And so we want to understand, do these change over time? Does one population that has a specific mutation come back more frequently than others? And again, this is to help us understand the natural progression of this disease under therapy. And so I'll highlight just a few here. And we can, again, think of this from the framework of what's happening in the recurrent tumor that's absent in the initial, and how might this be explained by therapy. So the first type we'll talk about is chemotherapy and how it changes those mutational patterns in a tumor. So shown here on the left is a plot of the data for each individual patient in our study and the number of mutations in their genome at the initial diagnosis on a log scale, and also on the y-axis, the recurrence. And we would expect that if the mutations didn't change between the two time points, that each point would fall on this linear line, this orange line here. But what we see is that there's a subset of patients that are mostly treated with this alkylating agent that develop hypermutation. So a large number of new mutations at recurrence versus their initial glioma. But note that not all patients that are treated with this therapy develop this hypermutation phenotype. So it's really just a small subset of patients that go on to develop these large number of new mutations that may explain why their disease came back. And so we can look at the different subtypes of glioma and see how this may differ between the two. And what we noticed was that there was, even though we had a smaller number of patients in this study for IDH mutants, that whether a tumor was IDH mutant with this chromosomal co-deletion, so these are oligodendrogliomas, or the IDH mutant without them, the astrocytomas, that they were more likely to develop this hypermutation phenotype following treatment with alkylating agents versus the IDH wild type tumors. So this is something that's interesting to us because, one, these patients aren't often as treated with these alkylating agents, but it's also interesting that not all patients develop this mutational phenotype. So we want to understand what is really driving this effect. So that was the chemotherapy angle for how the genome, the cancer genome, changes over time. What are some other types of therapies that patients receive? Well, one of the most common is radiotherapy. And so we see here in this graph of newly acquired mutations. So that is, again, the difference between the recurrent tumor and the initial tumor. We see that when patients either are not treated with radiotherapy or are treated with radiotherapy, that they have this increase in this number of small deletions. And these deletions are missing pieces of the DNA that, again, cause cellular defects and allow these cancer cells to continue to divide even in the face of treatment. But that we didn't see this when there's new pieces of DNA inserted into the genome, these insertions, or when these nucleotides or these DNA bases are swapped out for one another. So this radiotherapy phenotype that we see is really a deletion scar. And we're thinking in this latest research that we're thinking that these small deletions allow the cancer cells to better adapt to radiotherapy, leading them to be more aggressive. And so we can summarize the impact of treatment that I've talked about today. These are our two subtypes of IDH mutant and wild type. And each of these represents the proportion of patients being treated with these different therapies, this chemotherapy, this temozolomide, and radiotherapy, TMZ alone, radiotherapy alone, and the type of genomic change that occurs at recurrence. This includes the hypermutator phenotype or hypermutator profile I mentioned earlier, this small deletion scar appearance in the tumors, and also these this unknown category, or other category, which we haven't yet defined. And that is really a goal of our OPTIMUM project, is to better understand what are the shared changes that exist across patients. I'll note that also in the IDH wild type or the high-grade gliomas, that these genetic changes are less consistent, that there's fewer of them, fewer patients that share these genetic alterations. And so that we're actively interested in also understanding this other category for that tumor subtype as well. But anyways, why do we really care about these genomic changes? What effect does it have on patients? Well, we know that from our initial analyses that these treatment-associated genomic changes that occur only in a subset of patients are associated with shorter survival when including adjustments for other confounders. And what this tells us is that these treatment-associated genomic changes really reflect therapy-resistant mechanisms by the cancer cells that allow them to become more aggressive even when they are being hit with these therapies that are intended to kill them. And so really that is a point of our study, is to understand how these tumor cells are able to come back in some patients and lead to a recurrence. And that leads us to our current study, which is the OPTIMUM study, which is to build on that past research and actually engage with patients in order to help them, provide them with this feedback, this molecular feedback. And that is through this ability for patients to send in identify their samples at the initial and recurrent disease, send them to our central resource at Yale, and then we will perform sequencing to not only return results to patients about their sequencing data, but also incorporate this into a larger study where we will look across many patients to understand what are the common patterns that tumors evolve in the face of therapy, or how they change in the face of therapy, so that we better understand treatment resistance in those patients. And this is led by Dr. Elizabeth Klaus at Yale and Brigham Women's, Dr. Bethany Kwan at the University of Colorado, and Dr. Roel Verhaak at my institute, the Jackson Laboratory. It's important to note that our research is funded by the National Cancer Institute. And so what our broad goals for the study are to study that genetic evolution in the population that we haven't studied as intensely before, which is the low-grade gliomas. And what we're doing is we're studying these tumors at those two time points of initial and also a recurrence disease time point in about 500 patients to better understand what are the common paths that exist. And our goals are to identify genomic patterns of treatment resistance, ultimately to guide better treatment in terms of both the type and timing of treatment for low-grade gliomas, and also potentially identify new targets to prevent these tumors from recurring in future patients. So lastly, how do you join this exciting new study? Well, you can contact our group at glioma@yale.edu to express your interest. There's also plenty of links online, including at the American Brain Tumor Association, the National Brain Tumor Society, and also the IBTA. Their websites will have information about how you can join the low-grade glioma registry. You could also stop this video or even continue watching and scan this QR code, and that will bring you to the front page of the low-grade glioma registry. You can also simply search this in your Google search engine, and that will bring you to our registry. So I want to thank you today for taking the time to watch this video and learn a little bit about the research we're doing in low-grade glioma. And we'd be excited to engage with you in participating as a member or a participant in our study. Thank you so much and have a nice day.